thank you everyone for joining this webinar. We're going to talk a little bit about sperm and egg quality assessments, and what we kind of hope to do throughout the talk is just provide a toolbox of devices and assessments that can be done for your species and for the gametes that you collect. Next slide, please. So I think to start, it's always good to address why it's important to do your gamete quality assessment. And quality assessments do have kind of a variety of applications, starting from anything like assessing the health and reproductive status of the individuals in your care and the species that you work with, such that you might be able to assess things like infertility, or why you might not be obtaining the offspring you were expecting to, or anything like that. But it also has some other really great applications for kind of understanding the status of your gametes before performing things like artificial fertilization or developing protocols for cryopreservation, because you don't really want to preserve any gametes that are suboptimal. And so it's always good to have a baseline and understanding of what your gametes look like, and if they're viable, before doing anything like storing them for the future or performing artificial fertilization. Next. So a common theme that's going to kind of be popping up here and there throughout the whole talk, I think, is just how important it is to know kind of the natural history and know about the species you're working with. And that's everything from just gamete development, when they're going to be developing the eggs, when they're going to be developing the sperm, so that when you can retrieve them, whether or not you need to use something like hormone induction to help, obtain gametes for AF or cryopreservation. Retrieval, again, is species specific. So whether or not, there are different like protocols and things you can do that are highly species dependent in order to obtain your gametes, like obtaining spermic urine or egg stripping, as well as just any sort of natural release. Intestis macerates also is a way that you can obtain sperm, and this is more of an opportunistic method if you ever unfortunately have a dead animal, or if you're able to humanely euthanize your animals to obtain their gametes. What just happened? Anyway, I'll just keep going. But after your gamete retrieval, again, it's going to, hold on, I'll just let it load. There we go. It's going to be dependent on what you want to look at and what you're able to get from your species to determine what kind of assessments you want to do. And eventually, obviously, you would want to be able to use your gametes for either temporary or long-term storage, and eventually artificial fertilization, I think, is kind of the long-term goal, proof of concept. So obviously for today's talk, we are going to focus mostly on the different quality assessments you can do for gamete viability. Next. So the talk will kind of be divided into two segments. The first, we're going to be talking about the different sperm assessments you can do, and these are everything from highly basic assessments like concentration and motility to a little more advanced, looking at osmolality and pH, to even more advanced, looking at the morphology of your sperm, what it's supposed to look like, if there's any issues, and the functionality. So whether the sperm is working like it should. And then we'll also address some of the egg assessments that might be available for amphibians. There aren't a lot that are documented in the research for amphibian egg quality assessments, but I know of a few that we do in our program, and these are kind of the basic ones that can be seen in the literature, starting with highly basic assessments, just like visual assessments, egg measurements, getting a little more advanced into looking at the composition of the egg, what it contains in terms of nutrients and protection, like the jelly layers. Next slide. So as I said, we'll start with sperm. And I think it's kind of important to always start with just how you obtain the sperm, because it is species specific and dependent on what you're able to obtain. So the first method that I've kind of prepared a little video for you, our testes macerates. Again, this is a lethal method of obtaining your gametes. So it could be opportunistic, or it could be part of your plan just to be able to assess fertility or what's going on with your species in terms of sperm production. And it's a pretty simple process. You pretty much just take the testes and pull them apart or cut them apart in order to release the sperm inside. And then you'll aliquot it out, or perform any assessments you want to do, preserve it, use it for artificial fertilization, dependent on what you need it for. And then the kind of secondary way, that's a non-lethal way to obtain your sperm, would be through obtaining spermic urine. And this is where we get back at, just how important it is to know the best method for obtaining sperm from your species, because it is different. It's highly species dependent. And there are a variety of protocols you can use. These are probably not all of them, but just the ones that are most reported. And that's, scaring your animals into releasing their spermic urine, which doesn't work for everybody. Catheterization, and kind of forcing the spermic urine out, which includes palpation, cloacal lavage, and in some cases, passive collection. So, for example, for my species, the mountain yellow-legged frog, they really do not release their sperm easily. It's very, very difficult to get spermic urine from them. So I do have to use catheterization. I have tried a passive collection method, but I have not perfected that protocol. Next slide. If it wants to. And this is just another example, a different example of how species specific it is in obtaining your sperm. This is a great story that Rose actually gave to me. She gave me this picture. But these frogs are actually burrowing frogs from Australia. They live in very cool parts of the rainforest, mostly underground. And so all of these aforementioned protocols for obtaining sperm, like the heavily manipulating protocols, just really did not work on this species. And so Rose and her colleagues actually came up with this method where they just placed these frogs into falcon tubes with a little bit of liquid, and placed them near ice packs to keep them cool, and collected the sperm that was passively released into the liquid after the period of a couple of hours. And this was really the only method that worked to get sperm from these animals. So highly, highly important to know which species you're working with and how they're actually going to give you the sperm to be able to look at. Next. So now we'll kind of diverge a little bit because I want to talk to you about the different devices that you can use to assess your sperm. This is not a comprehensive list. It's just some of the more common ones that you see in the literature and the more common ones used for amphibian gamete quality assessment. And so you can go to the next slide. I've tried to kind of prepare it in terms of how much time it would take you with this device, how much money it costs, how much sample you need, and how much information it gives you, because each device kind of does something different. And these devices are going to be highly dependent on, or whether you use these devices is going to be dependent on what you need them for and what you can use. For example, the hemocytometer and the macular counting chamber both give you things like concentration and motility, but that's pretty much it. They do have a really low kind of initial investment cost. They're not that expensive to use. They also don't take a lot of time to set up. Their protocols are really straightforward and really easy to use. They do take a lot of time because you have to manually count the cells within these devices. So that can really take a good chunk out of your day, especially if you're looking at multiple species or multiple samples, or if you want to get, duplicate counts or anything like that. You can get a little more advanced and use something like an automated cell counter. It's a little more expensive than these options and takes a little bit less time. It is pretty easy to use, but it really only gives you concentration in most cases. So might not be the best device to use. And then you can get more advanced. This is kind of where you can start looking at these more kind of microscopic and advanced sperm quality characteristics when you start using things like the fluorescence scope, which unfortunately is quite expensive, usually has a pretty high initial investment in both cost and time, because you have to develop all of your protocols for the stains you want to use. And you still actually have to manually count your cells in the fluorescence scope. And so it does take a bit of time to both learn how to use and to both work it, but it can give you a lot of information, because you can start using all these different stains that Rose is actually going to be discussing in order to assess, the functionality and morphology of your sperm, as well as stuff like concentration and motility. And then you get really advanced and really expensive with some of these other devices like the flow cytometer and the CASA machine. These also have a pretty high initial investment in both time and cost, because you still have to develop protocols for stains. You still have to kind of develop protocols for the machine itself and make sure you know how to use it. So it is kind of this high initial investment, but once you do get it down, they can both give you just a wealth of information while using not very much sample and not taking that much time. So, I could put just a little bit of sample in and see all my live sperm, dead sperm, acrosome stained sperm, everything just in one shot, versus having to do it all separately and counting individual cells. So, I think they're awesome machines. There's just a wealth of information that you can obtain from them, but they might not be perfect for everybody either, because, you're not going to be able to bring a flow cytometer out into the field. They're pretty heavy. I don't know how heavy, maybe like 500 pounds. So you're not going to cart one of those out in the field to assess your sperm or anything like that. So this is kind of just a range of devices, and it's all dependent on what you need to look at, what you want to look at, and just what you're able to accommodate. Next slide. I thought this was a great figure to kind of lay out the different types of assessments that you can do. This isn't amphibian sperm. It's actually from a paper on human sperm, like quality assessments. But a lot of these tests can actually be done with amphibian sperm as well, starting from kind of these basic macroscopic tests where you're looking at, how much volume you received, what the pH was, what the osmolality is, and everything about, the sample that you were able to obtain. And then it gets a little more, in depth with the microscopic assessments that can be done, where you're looking at things like concentration, motility, whether it's moving forward or just wiggling, morphology of your cells, whether they look good, or if they might be morphologically, not intact. And then you get into the way more advanced kind of assessments that can be done. These usually, again, have to use things like the fluorescence scope or the flow cytometer. And it's where you're looking at, mitochondrial function, whether your DNA is fragmented, whether your membrane's intact, etc. The next slide. Thank you. So I'll start off with just kind of talking about some of the basic morphological assessments that can be done with your sperm. And I think this was a really great representation of the idea that you need to know what your species looks like first before you can make any sort of comparisons. Because this image, this black and white image on the left here, these are all different species of amphibian, and you could just see just the kind of huge amount of diversity in overall morphology, just in, head shape, tail length, tail shape, acrosome, etc. So it's really good to know what your sperm is supposed to look like before you can make any sort of morphological assessments, which is also kind of reiterated in this picture on the right, where you see the typical sperm, what the typical sperm would look like for this particular species, and then you would see examples of sperm that has a fragmented tail, or an abnormal head, or something like that. So this is kind of the first step, is just looking at your sperm overall morphology to make sure that it's all intact and actually looks good. And then you kind of get into some of the more advanced assessments where you can start looking at the specific structures or the functionality. And so from here, I'm going to actually let Rose take over, and she's going to talk about some of the more in-depth stains and procedures you can use to look at some of these other characteristics.
Thanks. So I think Leah's given us a really good background into why some of these things are really important. And I'm going to start at the more broader scale. If you remember the picture she showed of the sperm with the different levels of information we can get. And I find that this is a level of information that maybe sometimes we neglect to collect information for, but it's really important. So things like osmolality, pH, volume of urine for sperm, stuff like that is really important. And one example, you know, like Leah said, it's really important to understand the biology of your species. And so one example of that from a really long time ago is actually Sidney Ringer was a scientist a long time ago who was trying to do experiments with the heart of Arana species. And essentially he was trying to get the heart to beat without blood. And so he had been making saline solutions with distilled water, and nothing was working for him. And then one day, all of his experiments started working, and his lab assistant actually admitted that he had accidentally made up the saline solution that day using tap water instead of distilled water. And when he reverted back to the distilled water, the experiment stopped working again. And so when they looked into the composition of the water that they had used, they realized there was calcium and potassium and all of these wonderful things in there. And that's how we got the original Ringer solution that led to the simplified amphibian Ringer that many of us use in our research today. So stuff like that is really important, taking the osmolality of your sperm, but not just your sperm. Like if you opportunistically have an animal that died, like taking osmolality of blood, anything is all really important information that you might need one day. And osmolality especially is tied very closely to motility for amphibian sperm, as I'm sure you all know. So yeah, that's really important. So I just wanted to start with that. And I mean, sometimes you don't have enough volume or enough sample. So it really comes down to, you know, what your priority for the study is and what your goal is, what you're trying to find out. But I would encourage everybody to try and take this sort of data where possible. So moving on to motility, sorry for the resolution of this. This video is actually taken on a DVD player, would you believe? So this is an example of a Latoria species from Australia. That's a tree frog. And many of what we will be saying today ties back to what Leah keeps saying about knowing your species. So this is typical motility for a Latoria species tree frog, and you'll see there's lots of forward progression. There's a few sperm cells that are just moving in place, and I think there's one in the middle there that's not moving at all. And that's a really common way of measuring motility that you see in the literature. But there are also species of ground frog in Australia where you don't get a lot of forward progression motility, and it doesn't seem to be as important. So it's really important to know what's normal for your species. And here's another example of that. I think Ruth mentioned in a talk she did in this webinar series just recently, a salamander is not a frog. And boy, have I discovered that this year. I thought it would be really easy moving from being a frog researcher to working with the axolotl, and everything's different. So this is sperm from an axolotl. You'll notice it's spinning in circles. This is typical forward progression for this species. And these sperm are huge. They're like 100 micrometers in length, which is just, yeah, it's big. So something that I've discovered working with these species is that you need a little bit more room on a slide. So the typical methods that I had used to measure motility with just a drop on a slide and a cover slip don't quite work in this case. You need something like a 20 micrometer depth chamber, or the hemocytometer that Leah's already mentioned. And then moving up that scale of expense in equipment, you've got the CASA, which is computer assisted sperm analysis. And so what's going on here is this is sperm from a Xenopus laevis, and you'll see the red track there that's tracking the progression of the sperm across the screen. And then you've got like these different yellow dots in the background where they've picked up sperm that might be in a different class, maybe like non-motile or something like that. So this is something that you need to invest a fair bit of time initially to get your settings right. And so this can give you a wealth of information, like Leah said. So it can give you like a whole bunch of different parameters like velocity in a straight line and curvature and all these other weird and wonderful measurements that can just give you a lot more than like your standard count can do. But yeah, it's definitely worth looking into if you have that sort of technology. So moving on to membrane integrity, a lot of people call this, live dead staining. What it's really a measure of is whether the membrane is intact or not. And we tend to equate an intact membrane to a live sperm and a not intact sperm to a dead sperm. And so one commonly used stain, I'm going to start with the light microscopy. So these are things that you can do with a standard microscope. And then we'll move into some of the fluorescent methods. So this one here is a commonly used stain. It's called eosin nigrosin. And so basically the eosin will penetrate cells without an intact membrane, and they'll stain those cells pink, like on the right of that photo. And then the other cells will exclude the stain. And basically the nigrosin in this case acts as a counter stain so that you have that purple background so that you can see the intact sperm quite clearly there. But you can use eosin Y on its own. You don't always need the nigrosin. You just sort of need to play around with recipes and what works for your species. So then you can use something like trypan blue. So I find that trypan blue, depending on the optics of your microscope, can sometimes be a bit difficult to look at if the sperm is not like exploded, like the one on the left there that's clearly taken up the stain. But again, I think some stains just work really well for some species and maybe not so good for other species. I preferred eosin Y when I've been playing around with them before. But the principle is the same. The trypan blue is taken up by sperm cells with not intact membranes, and so you get that blue stain. And then the one on the right there has excluded the stain. So then fluorescence stains can be really, really useful. As you can see, it's like really clear. You've got this bright green versus bright red. And so those green ones are stained by cyber-14, and those are considered intact, whereas the red ones have taken up proteium iodide and fluoresces red. So this can take a little bit longer to prepare a sample because you need like these different incubation times with each stain. And you'll sort of have to optimize that staining process a bit more. But it can lead to really clear results, like as you can see there. And then some other options. I haven't tried these myself, but there are amine reactive live dead stains. They come in a whole bunch of different colors, and they're often used with flow cytometry, which is another benefit of using a fluorescent stain. You can use a flow cytometer to sort of count like way more cells than you can do manually, basically. And so you get this like more accurate proportion. So this comes in a whole bunch of different colors so that you can mix and match with different excitement. Like you could match like a green stain that is at one end of the spectrum with a different colored stain for something else at the other end. And that's how you get multiple types of data from one sample, like Leah mentioned. And so so basically the stains, the amine reactive stains, as I understand it, will stain all of the sperm one color. But if a sperm has lysed, then there'll be more amines from the middle of the cell available, and the stain basically reacts with those and causes a more intense fluorescence. And apparently these are fixable too. So there's it really just depends what you need to do. Fixable was quite attractive to some people because you don't have to analyze the sample then and there. So then moving on to acrosome integrity, this is a picture of a method that I've used. And so this sperm picture might look a bit funny to some of you, but basically you've got a smear of sperm across a slide, and then you air dry it and fix it in methanol. And so the sperm cells are actually dead, but fixed on the slide. So that's why morphologically they look a little strange, but the green tips on the end there are the acrosomes, which are preserved very well by the methanol. And so by staining them with peanut agglutinin, or PNA, conjugated to fluorescent FITC, which is the green color, you can stain the acrosomes. And then by counterstaining with something like rubidium iodide or DAPI or Herxt, you can see the cells, like the one on the top left corner there that doesn't have any acrosome, but you can still see there's a sperm there. And so by that way, you can count the ones with acrosomes versus the ones without. Then there are some other non-fluorescent options, like this Pope stain, which is a similar process for preparing the slide, but you don't need all of the fluorescent staining times, and you don't need a fluorescent scope, which, you know, can be quite expensive. And then there's things like Kamasi blue that are really good for just morphology in general, but you can also see there on the left, there are a couple of sperm there with that sort of dark blue tip, which represents the acrosome. And then on the right there, there's one with sort of a damaged or not intact acrosome. A couple of other options, I don't have any photos, but you could use Giemsa, which is commonly used with mammals. And then I've heard of this one called Spermac, which is like a commercially prepared one, which I think uses a different type of eosin and fast green. So that's not fluorescent. So that could be, both of those are non-fluorescent, and you can use them with a light microscope. So then drilling a bit deeper into some of the other functions of the sperm, the mitochondria are obviously very important. And it's been an observation of mine that after cryopreservation, for example, you might have a higher proportion of intact cells than motile cells. And so that sort of leads me to think that maybe the mitochondria are quite vulnerable during that cryopreservation process. And so if at all you have enough sample to start looking into stuff like this, and sort of from this point onwards in the talk, there's a lot of things that have been quite deeply investigated in the mammalian literature and other, you know, taxa, because it's been a lot of money and effort put into those areas, whereas amphibian sperm research is a little bit more behind. But there's no reason that you can't sort of adapt some of these methods towards amphibian sperm. It'd be really great to see some of that starting to happen. And so in this case, this stain is called JC1. This is an example in bull sperm. And the stain actually changes the way it fluoresces based on the membrane potential of the mitochondria. And so in a healthy sperm, you'll see mitochondria with a high membrane potential. And the way that the stain actually like has these J aggregates that accumulate depending on whether there's a high membrane potential or not, and that changes the fluorescence. So in the case of a high membrane potential, you get that red fluorescence that you can see on the right, and on the left, you get the low membrane potential, and then you can get this sort of orange color in between as well. So then you can also, if you're just more interested in visualizing where the mitochondria are, or whether they're, you know, maybe they've been sort of cleaved off during cryopreservation, you can use something like Mitotracker. So there's Mitotracker in a few different colors. This is an example from a paper with Xenopus sperm. You can see the green fluorescence of the mitochondria. I don't know if you can see my cursor, but it's just here at the bottom of the head. And then another example with buffonid sperm. So you can sort of see what I mean about the mitochondria in amphibian sperm is often in this sort of vesicle, like little bubble, and I think that leaves it quite vulnerable to damage. And so there's, I don't know, but that's just a theory I have. But anyway, you can see it's not in color, but you can see the fluorescence of the mitochondria green on the left, and then you can see the light microscopy version there on the right. So then looking at DNA damage, something like sperm chromatin dispersion can be quite useful. And so in this case, you can actually do this non-fluorescently, and you get this, basically you end up with some sperm with these halos, which are this kind of like pale pink color coming out. And then some of them over here don't have the halo. And so the ones with the halo are considered to be like intact DNA, and then the ones without the halo are considered fragmented. And so there are some other different sort of DNA damage methods in the literature that could also be helpful. Like I've heard of one called a comet assay, which I think is fluorescent. But yeah, it'd be nice to see some of this come into the amphibian literature in coming years. So for the last few slides I just want to talk about some other functionality considerations. So one thing is reactive oxygen species can be quite harmful to sperm. And when you put a sperm in a distressing situation, such as cryopreservation, you're basically causing the sperm to use up a finite amount of sort of, oh, I forget the name of sorry guys, it's basically a thing within the sperm cell that absorbs these reactive oxygen species. And then once they run out of that, the reactive oxygen species can start to cause damage to the sperm. And so you can use CellRock screen to look at reactive oxygen species within the cellular membrane. And then you can also use something like Mitosox red, which looks specifically at the mitochondria. And here's an example of that. Again, not in amphibian sperm, but basically from the left there, you've got mild to no reactive oxygen species build up. And then as the red fluorescence there intensifies as you go to the right, you can gather that there's a bigger buildup of reactive oxygen species. So then something like Reza's urine or an NTT assay can be used to look at like the metabolic activity of sperm. And so this is some data from honours student from University of Newcastle, Darcy Brett. And she's been starting to work up this as an assay for looking into the metabolic activity of sperm. And so basically you use Reza's urine, which when you add it to the sperm, some of it gets converted to ressorufin. And so that sort of reduction from Reza's urine to ressorufin, we hope can be correlated to sort of oxygen consumption and basically metabolic activity of the sperm. And so in this example, in this preliminary data, you can see that change in absorbance. So this is an absorbance assay. And so as that absorbance is reduced, that's indicative of ressorufin being reduced to ressorufin. So that's something that's being worked up at the moment, could be useful. And then just something that I've always wanted to look into but haven't had the time, just other important things for sperm function like calcium. And so looking at where the calcium is in the sperm and whether it's related with motility, this is another fluorescence thing that would be nice to see being looked into at one point. And I think this is where I hand it back over to Leah.
Thanks, Rose. So yeah, just kind of jumping ship here to talk a little bit about some of the egg quality assessments you can do. As I've said before, there isn't a lot documented in the literature on amphibian quality assessments, so this might not be a comprehensive list of everything you could do, but it's the kind of more common assessments that can be performed. And it starts with just basic visual assessments. So whether or not your egg is wrinkled or blotched or exploding in some cases. And it ties back into knowing your species as well, because it's important to know what your species eggs are supposed to look like. For example, these are two different species of Latoria frog, and as you can see, they're actually pretty vastly different in color. So, comparing these two egg images is not going to be very helpful because the color is going to be different. But if you knew what you were supposed to look at for your species in general, just to make sure that the eggs you're looking at are viable and look good, then that's going to be really important to know. Next. And to get a little bit more in depth with some egg quality assessments, measurements is a really easy one that you can do just to kind of address how developed your eggs are, and if they are viable. There's some research indicating that larger egg size is directly related to maternal investment, and usually larger egg size leads to healthier, more robust tadpoles in some species. So it's good to have a baseline idea of what your eggs are supposed to be in terms of size, but taking size, every year, every breeding season of your eggs might give you a good idea of, when you might have malnourished eggs or eggs that are not as viable. And then to get even more in depth with it, these are some of the more advanced things you can look at, such as basic egg morphology. So what the egg is comprised of, and this can be anything from, performing lipid assays to see what the egg contains itself in terms of nutrients for the, developing embryo, or what the jelly coating looks like. Egg jelly is actually extremely important for any sort of sperm egg interaction. So knowing, the size of each jelly layer, how many jelly layers your species is supposed to have, is going to be relatively important for understanding if there's anything wrong with your egg clutch itself before fertilization. And then as kind of a final proof of concept for actually both egg and sperm assessments would be to take any sort of fertilization data. So examining when your eggs cleave and if they are arrested in development at any particular stage, or whether they just are developing at all after they are laid. And that'll give you a good idea of whether or not, your pair was viable, or if something might be wrong with one side of, the gametes, the sperm or the eggs. And so this is also highly species dependent as well. Some species you can tell after a matter of hours if the egg is developing. Some, like the mountain yellow-legged frog, takes 24 hours. So it's a much longer progression. And, personally, this is actually one of my favorite parts of working with amphibians. I find this development really just pretty incredible and really awesome to watch. And so we did throw a little video clip of a developing embryo here, and it's just, man, it's just pretty cool. So I wanted to just kind of throw that in at the end so that everybody could be as amazed as I am every year when I get developing embryos. And so just kind of to wrap it all up, everything throughout this talk is just, it's a list of devices and assessments that can be done. It's a toolbox of things that you can do. You don't necessarily have to do all of them. Sometimes it's not possible to be able to look at everything, but at least this hopefully will give you an idea of some of the basic to advanced things that you can look at. And maybe it'll get you excited about, looking at some of these more advanced assessments that haven't even really been assessed very well for amphibians. I know it does for me. So hopefully this just gives you a great idea of what devices you can use, what assessments can be done. And And yeah, I guess that's pretty much it. Rose, did you have anything to add?
I think that's perfect. I think it's just one of those things where, again, it just depends what your goal is. So if you're trying to assess like fertilization capacity, then maybe like acrosomal staining is quite important. But if you can take data, I would always take as much as possible, because you never know what it might be useful for. So like, yeah, hopefully the toolbox is helpful, and if anybody needs any advice or help with it, then please feel free to reach out to either of us. And if we don't know, we can probably put you in touch with someone who might have a better idea.